Ribosomes are these molecular machines that carry out translation to make polypeptide chains. And ribosomes consist of both proteins and ribosomal RNAs, and therefore they're also called ribonucleoprotein complexes. Now, the prokaryotic ribosome is composed of two subunits, the large subunit and the small subunit, and their sizes are measured in what's known as Swedberg sedimentation units, and that has to do with centrifugation. So please do keep in mind that they don't add up. So the 70S ribosome is composed of the 50S large ribosomal subunit and a 30S small ribosomal subunit, but 50 plus 30 is 80 and they don't add up. Now, the large ribosomal subunit is made up of two ribosomal RNAs, the 23S and the 5S, and a total of 31 proteins, whereas the small subunit of the prokaryotic ribosome is composed of the 16S ribosomal RNA and 21 other proteins. The structure of the eukaryotic ribosome is quite similar, although on the whole the ribosome and the subunits are larger. So there is an 80S. The ribosome is 80 Swedberg units, and there's a 60S large subunit and a 40S small subunit, and there are 3 instead of 2 ribosomal RNAs, and a new one is a 5.8S ribosomal RNA, and there are more proteins involved. And the same goes for the small subunit of the eukaryotic ribosome. There's an 18S ribosomal RNA with 31 proteins. Here is on the left is an X-ray crystallographic structure of the ribosome, and you will see that at the interface between the large subunit and the small subunit there are 3 sites through which the messenger RNA passes through, and in this image the ribosome is moving to the right and the polypeptide chain exits through the exit tunnel. Now, what are these 3 sites? So there's A, where the amino acyl tRNA, the charged tRNA, enters the ribosome, and so this is the amino acyl site. Then P refers to peptidyl, and in the large subunit, this area is what would be considered the active site of the ribosome. It's called the peptidyl transfer center, and this is where the main action of making the peptide bond happens. And E refers to exit, and this is where an uncharged tRNA leaves the ribosome. So there are 3 phases of translation, there's initiation, elongation, and termination. Now, prokaryotes and eukaryotes differ slightly in how they initiate translation, and so we will discuss initiation separately, the prokaryotic case as well as the eukaryotic case, whereas elongation and termination are largely conserved between prokaryotes and eukaryotes. Now, the first step in prokaryotic initiation of translation is for the ribosome to actually find a messenger RNA. And this is a job that's performed by the small subunit, which will find and bind the messenger RNA. Now recall that if you have an mRNA, it will have a 5' end and a 3' end, and not all of the messenger RNA is translated. So you have a start codon somewhere in the middle, and then you have the stop codon somewhat later on, and this region on the 5' end that's not translated is known as the 5' untranslated region, UTR, and this region, the other region at the other end of the transcript that's not translated, is known as the 3' UTR. And it's the 5' untranslated region that plays an important role in helping the small subunit of the ribosome in finding the mRNA. This the 5' UTR has the sequence AGG This sequence is known as the Shine-Dalgarno sequence. Dalgarno. And this sequence is complementary to a region of the 16S ribosomal RNA, which is part of the small subunit of the ribosome in prokaryotes. And the small subunit recognizes or binds to the messenger RNA by Watson-Crick base pairing between the 16S ribosomal RNA and the Shine-Dalgarno sequence, which is located in the 5' UTR of the messenger RNA. You also have a few proteins called initiation factors, IF1, IF2, and IF3, that help in the process of initiating translation. And so first IF1 and IF3 bind the 30S a small subunit of the ribosome, and IF2 brings the N-formyl methionine. So it's a modified form of methionine, and recall that methionine is the start codon. So the first amino acid in every polypeptide chain is methionine, and in this case initiation factor 2 brings N-formyl methionine, or FMET, also called the initiator, and the mRNA, so that at the end of the process you get the small subunit bound to the mRNA, and the FMET, N-formyl methionine, bound to the start codon along with initiation factor 1 and initiation factor 2, and together this is known as the initiation complex. At this point, the large subunit binds, and initiation factor 1 and initiation factor 2 are released, resulting in a complete 70S ribosome with the methionine in the peptidyl site, the P site, base paired with the AUG start codon. Now, in initiation of eukaryotic transcription, the recognition of the mRNA by the small subunit is not carried out by the Shine-Dalgarno sequence, but instead by the 5' cap. And you have initiation factors, just like in prokaryotic initiation, and you have a charged methionine tRNA. In this case, it's not an informal methionine, but just a regular methionine, and the small subunit is already bound to the methionine and then recognizes the mRNA, whereas the order was reversed in prokaryotes, where the mRNA was bound first, followed by the arrival of the methionine, and then the mRNA, the start codon, is introduced into the P site of the ribosome, followed by the binding of the large subunit, resulting in the formation of the ADS initiation complex, which is the whole ribosome with the mRNA placed in the right location, which is the start codon in the P site, the peptidyl site of the ribosome, along with the methionine, where the anticodon, methionine tRNA, where the anticodon is base pairing with the codon. Now, elongation, which is largely conserved between prokaryotes and eukaryotes, this elongation factor TU, brings a charged tRNA, and it enters into the A site. At this point, the ribosome catalyzes the formation of the peptide bond, and the growing polypeptide chain is transferred to the new tRNA that has come in the A site. At this point, translocation occurs with the help of a elongation factor G, which pushes the two tRNAs to the left, so that the first tRNA is now in the E site or the exit site, and the second tRNA is in the P site or the peptidyl site, and elongation factor G occupies the A site. At this point, EFG leaves the A site, and the ribosome is ready to accept a new charged tRNA in the A site. And once EFDU brings in the next charged amino acid tRNA, the uncharged tRNA in the E site leaves the ribosome. Translation termination occurs when a stop codon enters the A site, and at this point, this stop codon is recognized by these proteins called release factors. So RF1 recognizes the stop codon UAA, and when that happens, the ribosome disassembles, and the uncharged tRNA is released, as well as the polypeptide chain and the tRNA that was in the P site of the ribosome. There are three release factors. RF1 recognizes the stop codons UAA and UAG. Release factor 2 recognizes UAA and UGA, and release factor 3 helps in the process. The way the stop codons are recognized leads to an interesting consequence, where you can have suppressors of nonsense mutations. So let's say I have an mRNA with some sequence GGG, CAG, G, AG, AAG, and 3'. And this codon GAG is for glutamate, and let's say we have a charged glutamate tRNA with the anticodon loop CUC recognizing our GAG codon, and we have a glutamate amino acid on this charged tRNA. Now, in a nonsense mutation, you might have this G, let's say, mutated to a U, and then which changes the GAG to a stop codon, and now instead of the charged glutamate tRNA being able to recognize this stop codon, you will have release factor 1 that recognizes this this codon, and therefore causes premature termination of translation, and that results in a truncated protein, resulting in a mutant phenotype of the nonsense mutant. However, if there is a mutant glutamate, okay, so we have a mutant glutamate tRNA, where in the anticodon loop, instead of having CUC, we have AUC. Okay, since tRNAs are also synthesized, are coded for by genes, those genes can have mutations, and in this case, there is a second mutation in this organism, where in addition to the G mutating, being substituted by a U in the gene of interest, the tRNA gene has a mutation, so that instead of having CUC as the anticodon, the glutamate has now, the glutamate tRNA now has AUC as the anticodon. And of course, if that happens, then instead of the release factor recognizing the codon, the mutant glutamate or rather the mutant glutamate tRNA will be able to recognize the stop codon. And what this means is that instead of terminating translation at UAG, translation will be able to proceed, and the mutant phenotype of the first mutation of changing the G to a U, which introduces a premature stop codon, will be suppressed by the second mutation, this one occurring in the tRNA gene, the gene that codes for the glutamate tRNA, which changes the C to an A and allows this glutamate tRNA to base pair with the stop codon, and allows a translation to proceed, and therefore suppresses the mutant phenotype. And a final situation that's possible is that of imperfect suppression, and that can happen if we have a situation like this, where instead of the glutamate tRNA being mutant, it's, let's say, the lysine tRNA that's mutant, and lysine's anticodon is UUC. So let's say there is a mutation that changes the lysine anticodon to AUC. So now we have a lysine tRNA that can base pair with the premature stop codon, allowing once more translation to proceed, but resulting in a protein in which the glutamate has been substituted by a lysine. This can still cause suppression, because the effects of a nonsense mutation where you get a truncated protein are usually going to be much more severe than a substitution of one amino acid residue with another. And therefore, this situation will result in suppression, but the suppression will be imperfect.